Good morning, gentlemen and ladies. It is good to be with you. It's always good to be with the people of God. And this morning, as I was doing my devotional, it dawned on me that one day even death will die. On one day, we won't have to worry about this because in the presence of God, in the presence of Jesus, there is no death. So one day, death itself will have to flee because the life giver will be with his people. That is, I think, our blessed hope. So as we struggle through these very tough times, I hope just that thought will give you hope and give you peace, even in the midst of the storm. My goal today is really just to share with you. I have some slides. I don't need to get through all of them. I just want to use them to set up a discussion, but really want this to be a way for me to answer the questions that you might have so that we can learn a little bit of science. And I'm going to share my screen with you now. Okay, so the virus, the vaccine, the variants. And so I want to have a little disclosure that I have no intellectual property interests in any of these companies that I will mention where vaccines are concerned. And it's also that it's not my goal to convince anybody to take a vaccine or anything like that. This is a very personal medical decision. It is, however, my goal to share with you the science behind all of this so that you'll be in a better position to actually make that decision yourself. And so I want to make that very clear at the onset that, my intentions are really from the scientific perspective. So let's start from the beginning. We've had coronaviruses around for a long time. We've known about them for a long time. In fact, the initial coronaviruses, the NL63, for example, was discovered in 1960s by professors at the University of Chicago. And for a long time since then, 1967, 1970s, more of them pop up. So that's these that you're seeing here. In fact, they were so innocuous that nobody bothered to give them real names. They were given these alphanumeric codes because all they did is that they caused the common cold. So it's very likely that in many past seasons you've had the common cold and it was caused by coronavirus. But 3 times now in the human history, we have had lethal coronaviruses that have jumped into the human population. These viruses live in bats, in rodents. And so in 2002, 2003, a coronavirus jumped from bats into the civet cat right there in China and spread to humans from the bats. And then we had human to human transmission, leading to about 8,000 and something people infected, about just under 800 of those died. And at that point, they started in earnest to try to find a vaccine, to make a vaccine against these organisms. Because in microbiology and infectious disease, we know if you have one jump, it's just a matter of time before you have the other. That's why we have influenza and all of those. It's what we call zoonoses. We're accidental hosts of these. But then that epidemic went away so quickly in 2004 that they weren't able to test a vaccine, even though they had started developing one. And then in 2012, another family member, the Middle East Respiratory Syndrome virus, or MERS, jumped from a bat into camels and is currently being transmitted from camels to humans. We don't know about it a lot here in the Western world because it's very common in the Arabian Peninsula. We don't have a lot of camels that we're interacting with. And then, of course, you know what happened in 2019 when SARS-CoV-2 jumped from bats, we know, probably had some other intermediate host and jumped into the human population. And that has given us what we have come to know as SARS-CoV-2, which is the name of the virus. The family of viruses, corona, describes the way it looks under the microscope. So this is a cartoon that you're seeing here of the virus. You have seen these cartoons around a lot, but here, this is what we're seeing on the microscope when we look at the virus itself. And you can still see those spikes. Each viral particle has about 100 of them. And these are important to the virus because this is actually what it uses to attach to our cells to get in. But it's from that look that the name corona in Latin means crown. And so COVID-19 is coronavirus disease 19. The virus itself that causes it is severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2. And I want to show you this image, and it's also the background that's behind me. This is what happens when one virus, coronavirus, infects a cell. And the cell is this purplish, brownish blob that you're seeing. The yellow dots and the green ones behind me in my background are actually what happens when the virus infects. So these are viral particles that have been colored for you. Because one viral particle within 10 hours can turn into hundreds to thousands of viral particles that exit the cell to infect new cells around it. So this is a rapid infection that you have. The experts believe that this viral jump happened somewhere in Wuhan, not necessarily in 2019. It could have happened before, but that's where this current outbreak started. It's a bustling city, lots of people going in and out right around the time close to the Chinese New Year. But it's also a place that we've known for a long time have what are called these wet markets. But in the 1970s, when China was having a huge problem with hunger and poverty, having trouble feeding its people, it started allowing people to catch and trade wildlife. And it started these wet markets where people come with live animals and they slaughter those animals right there. And these include everything from fish to bats to you name it. The wildest of animals, snakes, frogs, they're all slaughtered and sold there, traded there. It's a huge part of their economy, a huge part of the culture. And just to show you what happens in some parts, not all wet markets, some of them are very well set up and are well done. But here you have a chicken that is being sold, sitting on top of a basket of vegetables that's also being sold. And here you have frogs in this basket that are also being sold for food. They're slaughtered here if the customer so desires. The entrails, the intestines and stuff are in here. And here is a cutting board right up here that is used to slaughter and to cut up them up in pieces. Here's a basin, plastic container that's used to weigh it on the scale. And if you notice, nothing is really clean there. So it's not a huge stretch of the imagination that if you're buying food in this place, and if you notice, it's a mix of vegetables and all sorts of meats that they're selling there. And the thought is that it's just a matter of time before you would have had this jump. And so what is resulted in is today we have 136 million cases of recorded coronaviruses around the world, over 2.9 million deaths worldwide, and keep going. In your neck of the woods in Canada, where you are in Toronto, it's the hardest hit province there, maybe because of the larger population, but you have over 386,608 cases as of a couple of days ago, with also the most deaths. You can see that the entire province of Ontario, it's trending up. So in Quebec, many of these other areas that are highly populated, you're continuing to have an increase in the number of cases there. So that the updates 2 days ago, you can see that the new cases, total deaths there, tests that are being performed. And so you have a percent positivity of 3.8, and that is not good for any populated area. I'll pivot quickly to say that the major pathology of the viral infection is lung disease. The virus can infect the airway. And if you look at this cartoon, you see when you inhale, the air, oxygen rushes down through the bronchioles into the lower part of the lungs where you have these little sacs, literally like bubble wrap sacs called alveoli. And it is here that you have exchange of oxygen and carbon dioxide in the blood that is coming from the tissues and oxygen that you've just inhaled. And that exchange happens here. When you have pneumonia, those sacs that are supposed to carry out that air exchange are filled with fluid. In fact, mucus is what you have in there. And as a result of that, makes it very hard for you to breathe. And if this continues, this continued inflammation can lead you to the hospital. And many patients, if it's not resolved in a short time, will have to be put on a ventilator where they have to force oxygen into your lungs because you don't have the open airways to carry it. And so essentially, here's a normal lung. And here are lungs that have COVID. And COVID is really severe because most times when you have pneumonia, it's in one lobe of the lung, one side of the lung. COVID has been known to be always bilobal. So you see this consolidation, all those white areas are areas where you can't have oxygen exchange because it's just loaded with the virus. The problem is that a lot of us think about COVID in a binary way, that you either live or you die from COVID. It turns out that third group there is a subset of people who have what is called long COVID. And these individuals are shackled with symptoms from fatigue to headaches to shortness of breath, hearing loss. In fact, people have committed suicide after recovering from COVID because of the tinnitus, the ringing in the ear that just won't stop. So actually living after having gotten COVID can be just as devastating for people. And the data shows that 1/3 of those with long COVID that continues months after you would have recovered and the virus is no longer being tested out, you're no longer testing positive for the virus. 1/3 of those individuals have psychiatric issues that manifest themselves. So a lot of issues need to be resolved. And here I'm showing you all of this red that you're seeing here, this brownish red, are the viruses, and they're right at the base of the brain, in what we call the olfactory mucosa. So although scientists haven't yet confirmed the presence of the virus in the brain, we can see it at the base of the brain. And no wonder people lose their sense of taste and smell. People also have what is called COVID tongue. You see these papular rashes that you're seeing here, these indentations on the tongue, the swollen tongue that you're seeing. These are all things that people who have recovered, so to speak, from COVID are having. And then more recently, we're seeing people with these gangrene of the extremities of the digits, fingers and toes. People are losing their fingers and toes. Some people have had their legs amputated after having recovered from COVID, because see, COVID caused clots, and those clots actually lodge in the small blood vessels, the capillaries and venules. That cuts off the blood flow to those areas, and they just die. They develop gangrene in those tissues. As we also know, people with comorbidities, people with hypertension and obesity and diabetes, and people with COPD and other chronic lung diseases and heart disease are more at risk for dying of this disease. And those who have who have more than one of these comorbidities are at increased risk. You can see 90% if you have two of these issues listed there. When you add to that the fact that we're not only dealing with the original viral strain that came out of Wuhan, that the more the virus replicates is the more it makes errors in copying its genetic material. And that is what gives us the variant. So as long as the virus is widespread in communities, like what is happening in the provinces there in Canada, you're gonna have development of these strains. So far, we have 3 standout strains, but there are certainly many more circulating. They just haven't taken over the way this B117 strain that was first discovered in the UK, the B135 strain from Africa, and the P1 strain in Brazil. These are now spreading in North America. In many places, it's the dominant strains. That is the case here in my state of Massachusetts, certainly in New York, and many places in the US. These strains are spreading, and you can see that they start in one place, but it doesn't take long before they spread to other areas of the world. The virus itself binds to our cells using these spike proteins, right? This thing that looks like crown on its surface. And these spike proteins look like this. They're a trimer. 3 pieces come together to form one. And what it does, it binds to this protein that we actually need called ACE2. This is angiotensin converting enzyme receptor that is used to balance our blood pressure. So it's indispensable. It turns out that a lot of cells in our body, in all parts of our bodies, have these receptors on them. And when you see this, when you see just the plethora of organs and systems in the body that have these proteins that weren't made for the virus to enter, they were made for physiological function, then you can understand some of the effects of this virus, some of the pathology that we're seeing from this virus. Because from the brain to the testes, to the placenta, the liver, the eyes, the pancreas, there are people now who go into the hospital for COVID. And when they leave, they turn up having to go back to the hospital. And guess what they turn up with? They turn up with diabetes. They never had diabetes before, but now they're having a COVID-related diabetes. They're having COVID-related hypertension because it seems as if the virus is able, either through the immune system or of itself, destroy the islet cells in the pancreas that produce insulin, for example. So again, I guess what I'm trying to say is getting infected with this virus is no good at all, whether or not you survive. But look at what these variants can do. And all that happens, as I said, you can see these black balls here. These black balls are just changes in amino acids. Proteins are made up of amino acids, right? And this is the spike protein, animated. One of my former immunology professors created this. And you can see these black areas here. It has these mutations. But what is important is right in this area. These purple balls show you where the virus actually binds to our cells. It binds to that ACE2 receptor. And if you notice, the virus has 3 amino acids that it has changed in this spike protein area of binding. It can't change many more in there, because if it does, then it won't bind anymore. It's like you can file your key down a little bit and it might still fit into the lock, but if you file it down too much, you won't be able to open the door, right? However, you can file it down so it fits more perfectly in the lock, and this is exactly what the virus has done. And let me just show you an animation of what happened in the UK at the end of last year. This is the B117 strain. They originally had the original strain, and this is the way the map looked. The darker it is, the more cases they have. And so take a look at that. In 1 month, the entire United Kingdom was overrun by this new B117 strain. It is more contagious. We know that it's about 50 to 70% more contagious, and it's now clear that it is more deadly. And that is what is happening now in Michigan and several other states in the United States. You can see the UK had gone down significantly in the number of cases that we're having until this B11 strain was found here. And look at that. It spiked right back up. So that is one of the worries that we have about this virus. Now, I want to tell you a little bit of story about Manaus, Brazil. Maybe some of you on here have friends and our family in Brazil. In October of last year, Brazil had experienced a really huge outbreak of this virus, and the original strain of the virus had infected over 76% of people in Manaus, Brazil. And for the first time, we saw an era where so many people had survived the initial outbreak that they were immune, essentially. They had herd immunity. So many of them were already exposed to the virus that they built up immunity that protected essentially the rest of the population. So we saw in real life this herd immunity in Manaus, and their case numbers went down almost to 0. They were not having any infections anymore. People weren't going to the hospital. And then something happened. You see this? This is right around January, end of January, February. The numbers started going back up, and so did the hospitalizations in Manaus. So what happened? That P1 variant in Brazil started coming up, and that P1 variant started killing people, so that Manaus, now, this is what they had to do. They had to clear a part of a forested area to make a mass grave so that you can have people being buried there from this second strain of the virus. In other words, these people were immune to the original Wuhan strain, but not to this P1 variant. That is one of the issues with this virus. So the World Health Organization says the 2 public health interventions that have had the greatest impact on the world's health are clean water and vaccines. And I agree. Here's the thing about how vaccines work. Sometimes people think that it's either your immune system or vaccine. Vaccines don't work without a functional immune system. In fact, if people are sick and they don't have a functional immune system, you won't vaccinate them because it just won't work. So here's what happens when you get exposed to any microbe, not just a virus. God created us with 2 arms of our immune system, innate and the adaptive arm of our immune system. Here's the thing. You don't want the adaptive arm to kick in every single time you're exposed to something. This is like calling in the army every time you hear somebody maybe breaking into your house. You don't call the army, you call 911 and you get your local police, or you call whatever emergency services and you get your local law enforcement to come and help. If you call in the army, number one, it takes a long time before they build up to deploy. And so essentially, it takes about 2 weeks before this adaptive arm of the immune response kicks in fully. So the innate arm of the system is supposed to hold down the viral load or the bacterial load, until it is deemed necessary to have this adaptive arm of the system come in. If the innate system can't do that, then the virus is going to overcome the system, because viruses replicate way faster than our immune cells do. For example, the coronavirus in 10 hours, as I told you. And so if you notice what starts happening, the moment the adaptive arm kicks in, then the viral load goes down. This is basic immunobiology. We've known this for 100 years almost. This is how the immune system works. So people sometimes think, oh, if I have a functional immune system that God created me with and I'm healthy, I don't need a vaccine. But here's the problem. This virus, as I said, takes about 10 hours before it turns over, before you have a full new set of progeny. So let's say you would start off with 1,000 viral particles. Within 10 hours, just 10 hours, not even half of a day, you started off with 1,000. You're going to have over 100,000. And by the end of the day, you already have over 1 million viral particles in your system. The immune system, the adaptive part of the immune system that is responsible for giving you long-term memory, because you see the innate part of your immune system doesn't have a memory. It protects you for the mere fact that you're a human. There's cells, there are barriers, there are mucus, there are enzymes that help to protect you and keep the bacterial load down. So here's the thing. We hear about antibodies and neutralizing antibodies a lot with this viral outbreak. The antibodies are these Y-shaped proteins that are produced by the adaptive part of the immune system. That's a second arm that takes longer to kick in. And that is actually what is able to neutralize very effectively these viruses. So when you have an exposure, which is where this red arrow is, when you have a first exposure to any pathogen, it takes about 2 weeks, 14 days. That's why your grandmother was able to tell you, if you have a cold, within 2 weeks, it's going to get better. They had figured that out, not knowing what the mechanism was, but it's because it requires the adaptive arm of the immune system. And you can see it doesn't kick in fully until there, and then it starts waning. Now look at what happens if you get exposed to that same thing again. It doesn't take 2 weeks anymore. Literally, it takes 1 day. And not only is it faster, but you get a much higher response. This is the reason why we vaccinate. This is why vaccines work. Vaccines work because you give this initial exposure through the vaccine. You prime the system. And so when you actually encounter the again, when you actually see the virus for the first time, it's really not your first time, because you've tricked the immune system into thinking that it already saw the virus. So it makes a response. So what would have taken 2 weeks now takes a day. And that's how you're protected. So vaccines do not prevent you from getting infected. They prevent you from getting sick and dying, because they have already primed the immune system so that the immune system is ready to respond. The second arm of the immune system is ready to respond. Now, there is absolutely no way to avoid this primary immune response. It doesn't matter how healthy you are. It doesn't matter if you're a vegetarian or you're a meat eater. It doesn't matter how long you have been healthy. You cannot avoid this, because this was put in for a reason. You don't want the second arm of the immune response to kick in if it's not necessary, because that is why you feel fatigued. That is why you feel tired, because when it comes in, it's a whole system. The entire body is having this activation. And you don't want that every single time you're exposed to a few microbes here, a few microbes there, because we're exposed to microbes every day, right? So no diet, no lifestyle, no supplement can make that delay any less, that 2-week delay. So that's why vaccines are in there. And I hope I've made that clear. And so now what happens is that when you're exposed to the virus for real, these neutralizing antibodies, these Y-shaped proteins, which I like to say are the cheap ammunition of the immune system, they come and bind to that spike protein, which is the kind of the key that the virus uses to unlock our cells to get in. It binds to it, and it's like taking duct tape and wrap it around your key. You can't fit in the lock anymore, so you can't get in. And so now you're protected. And that's essentially what you want to happen. So here again, if you have COVID, it comes in within a very short time. Look at how high the viral load gets. Your system becomes overwhelmed. And what we know that this virus does, it actually very effectively prevents that first arm of the immune system from kicking in fully. So by the time the adaptive arm kicks in over here, you're already pretty sick. In fact, most people get hospitalized within 6 to 8 days. But remember, the antibody levels for the adaptive system doesn't kick in fully here. By that time, you could already be dead from the virus. So without something to protect you, to give you that initial protection, you're in trouble. Here's the mystery of vaccines, right? Two boys, both of them infected with a smallpox on the same day from the same person. They're both the same age, 13 years old. The only difference, this boy here on the right was vaccinated, his friend was not, right? That is the effect of vaccination overall. So right now, there are 50 COVID-19 vaccines in phase 1 trial, which means that they're just testing safety in the first phase with humans, just about 50 or so humans in those trials, 35 in phase 2 trials, 23 in phase 3. So phase 2 trials, they're still just checking safety, but also efficacy. In other words, does it protect? And phase 3, as you know, is the bigger human trial with tens of thousands of people in them. And there are 5 vaccines that are fully authorized in different countries, 8 of them that are approved, like the ones that you have in Canada there, ones we have in the US, and 4 of them have been abandoned. So as you can see, a lot of people are working on vaccine for COVID-19. The ones that have been approved here in the US and in Canada, Pfizer, Moderna, and in Canada, what you have is the AstraZeneca vaccine. The Covishield is an AstraZeneca vaccine that was just made by another place. We have the Johnson & Johnson here that if you heard the news this morning, the FDA has stopped the giving out the Johnson & Johnson vaccines because of the 6,000 something doses of Johnson & Johnson that they've given out. They've had 6 cases of blood clots, very similar to what you've been having, what people have been having in Europe with the AstraZeneca vaccine. Turns out that they're made on similar platforms. These are what we call viral vector vaccines, where you use an innocuous virus to deliver the code, the genetic code to make the spike protein for SARS-CoV-2 so you can get vaccinated against it. So they're investigating these. These are very rare blood clotting events, but they feel that the risk, especially for younger women, which is where they're seeing it in people under the age of 50 and mostly females, that this is worth investigating. But let me explain to you a little bit of how the mRNA vaccines were made. So essentially, when you look at a cell, basic cell biology, a cell has 2 major compartments. This is a cartoon of a cell. This purple blob here is the nucleus. And this is where the DNA of the cell is. Your DNA is the code that codes for everything that the body makes. Now, proteins are the workhorses of the body. Pretty much every gene that you have in the system, you want it to code for a protein. It is the protein that you use to digest your food. They're amylase. And it's proteins that actually lead you to clot when you get a cut. In other words, proteins are what carry out the functions of the body. So the genetic code to make those proteins is in here. The factory to make the protein is out here in the cytoplasm. So how do you get to make proteins out here when the code for the protein is in here? What the body uses is called the messenger RNA, mRNA, a copy of that gene segment that is necessary to code for that protein, leaves the nucleus, comes through these nuclear pores and come out here where it's read by these ribosomes, these little dots here that you're seeing on the endoplasmic reticulum. And this is a continuous factory. In fact, it is the only reason why viruses are able to infect us and be productive, because viruses do the same thing. They come in and they use this factory to make all the proteins that they need, because viruses don't have the capacity to make their own proteins. So what these manufacturers did essentially is that they use the same technology as what they're seeing viruses doing and what the body already does. They have given us the mRNA encapsulated, and our body sees that messenger RNA, that copy. And the way I used to like to describe it, let's say that you are about to cook a very special meal for your loved one, and you want to use an original recipe that was created in 1780 something, but it's not widely found. It's not on the internet. It's actually in a book. This rare book that's actually making the rounds to different libraries. And it turns out that your hometown library is where this book is sitting at. And so you go to the library. You can't take the book out of the library because it's literally like a historic piece. They won't allow you to take it, but you can actually look at it and you can photocopy pages out of it. So you go to the library, you borrow this book on reserve, and you photocopy the pages with the recipe of that pie or cake that you want to make, and you take that photocopy out of the library. That photocopy is the messenger RNA. You now take it to your kitchen, which would be the ribosomes, the protein factory, where you have all the ingredients, and you make the cake out of that. So that's literally what this is. So essentially, what the vaccine is doing is that it's giving you a photocopy of the gene that is used. And I'm going to show you a little bit of how that actually happens in real time.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay. So as you saw there, this was an animation showing you based on what they've come up from the molecular tools as to what is happening. When that messenger RNA gets read and that protein gets made, 2 things happen. That transcript, that photocopy that was made, that messenger RNA gets destroyed, doesn't sit around, right? In other words, after you're done with the cake, you throw this thing away in the trash. It's almost like Mission Impossible. This message was self-destruct in 5 seconds, right? It's not exactly 5 seconds, but within minutes to hours, that messenger RNA is gone. The second thing is that that protein that is produced, in this case for the vaccine, it would be the spike protein, because all that scientists did is that they got the code for the spike, not of the entire virus. They got the DNA template from that, and from that they were able to make that messenger RNA in the lab. They had to encapsulate it in lipids. How do they know this? Because they've been trying to develop mRNA vaccines for the past 20 years. This is not a new technology. I've been teaching about mRNA vaccines in my classes for the past 10 to 15 years, right? So they've developed this technology. They realize that if they just put the mRNA by itself, the cell sees it and realize it's not supposed to be there and degrade it. If they put it in these fat droplets, these nanoparticles, then it will make it into the cell. It makes it into the cell reads it, right? Reads that transcript, make the protein that it codes for. Those proteins are released. The immune cells see them, respond to them by making this Y-shaped protein antibody, and that's how you get that protection, right? It's a priming of the system. So here's an animation.
mRNA technology has never been used before in an approved vaccine, but scientists are using it now to combat COVID-19. mRNA, or messenger ribonucleic acid, are snippets of genetic code that tell cells to build proteins. mRNA vaccines take advantage of that protein-building process to trigger an immune response and build immunity to SARS-CoV-2, the virus that causes COVID-19. Instead of using a small dose of live or dead virus like some other vaccines, mRNA vaccines use a tiny piece of the virus's own genetic material. Here's how it works. This is the virus. These are called spike proteins. Scientists have isolated the genetic code for those proteins, a set of instructions that are put into the vaccine so your body knows how to make them. Your immune system won't make an actual virus particle, but it will make copies of the angry red spike protein. In other words, no gray ball, but an angry red spike. Once vaccinated, your body starts making those spike proteins so your immune system will develop antibodies to fight the real thing, SARS-CoV-2, the virus that causes COVID-19. The Pfizer and Moderna vaccines that are created for COVID-19 will be the first licensed mRNA vaccines ever.
Okay, so that is essentially it. And as I said, the mRNA technology has been around for a long time. As you can see, 1992, where people were publishing about this, right? This is in the journal Science. And just to show you a few more, this is 2017. This is a phase 1 clinical trial trying to make a rabies vaccine using the same technology by Moderna. mRNA vaccine for flu shots, right? They were trying to make a flu vaccine using the mRNA technology, and so on and so forth. This is another Moderna trying to make a vaccine. And this is actually in 2020, just before we knew anything about the pandemic. And so again, it's not a new technology. In fact, you can see this is a huge Nature Reviews paper, mRNA Vaccines, a New Era in Vaccinology. And this again is a paper from 2018 before SARS-CoV-2. So again, my point is that this is not a new technology in terms of we're just figuring it out. Now, you heard the term nanoparticle there. And I know that a lot of people get anxious when they hear about nanoparticle, but all the word nano suggests is size, right? A nanoparticle is just describing the size of a particle. So a baseball bat is about a meter long, right? A dime is about 1/100th of that. And a millimeter is 1,000th the size of a meter. A micrometer is 1/1,000,000th the size of that meter, right? So that's micro here. Nano is 1/10000000th the size of a meter. In fact, a nano is so small that if, you know, those Hot Wheels toys that kids drive around, those Matchbox Hot Wheels. So the idea is that if each person on Earth was the size of a nanometer, everyone on the planet would fit into that Hot Wheels Matchbox car, right? That's how tiny a nanoparticle is. So here's what I hear all the time. People say, oh, you know, don't you know that Bill Gates and the government are putting microchips in these vaccines? Remember, this is micro up here, but remember the vaccine is delivered in nano. Which one is larger? You can't put a microchip in a nanoparticle because a microchip is that much larger, 1,000 times larger than your nano, right? So again, some of those accusations don't make sense. So what is in these vaccines? The vaccines, the mRNA vaccines have mRNA in it. It has salts. Salts as in saline is what is used to dilute it. It has these lipids. And lipids don't mean pork. These are fats that they use to encapsulate, and they use different combinations of them. A little bit of sugar, sucrose is there as a part of the buffer. That's what's in them. They don't even have any preservatives in them. This is for the Moderna and Pfizer vaccines. Do they cause side effects? They absolutely do. All vaccines do. So for the Pfizer vaccine, 84% of people who have it gets a rash at the injection site. I was one of those people who didn't feel any pain when I got this vaccine, but 84% of people do. People feel fatigue. I was one of those people. Headaches, muscle pain. People have these types of things. Same thing. So a few people have chills. It turns out that a lot of these effects, the fatigue, the chills, the fever, this is a part of your immune response responding to what you've just put in there. You know, you have millions, maybe trillions of copies of these proteins being produced, and the immune system is saying, hey, this is not supposed to be here. We're making a response against it. That's why you actually have these types of responses. Now, the AstraZeneca vaccine, Covishield, that you have there in Canada, it's made a little bit differently. Same thing you're making it against, the spike protein. What they do, they get that spike protein gene, but instead of making an mRNA from it, they actually put it into another virus, an adenovirus. Adenoviruses are cold virus, but this one is actually from a chimpanzee adenovirus. And so it doesn't cause disease in humans, and moreover, they have transformed it, they have changed it so it doesn't replicate, it can't make more of itself when it gets into your body. So essentially what they're using it as a Trojan horse, so it can carry in this genetic code. Once it gets in, the cell takes it up, when it's injected in there through the vaccine, cell takes it up, and it injects this genetic code into the nucleus of your cell, where it immediately gets copied into messenger RNA. That messenger RNA leaves the nucleus in the same way I showed you before, gets into the cytoplasm, where it's made into a protein. These protein pieces get assembled, and they get taken outside. And when the immune cell sees it, it picks it up. This is what immune cells do. It breaks it down into tiny little peptides, because the immune system doesn't recognize stuff and respond to it when it's this big. And essentially, eventually make antibodies that are able to bind to and neutralize the virus. So that's how that is made overall. So in Canada, what we're seeing is that people are having different side effects. Vaccination site pain, you can see, is the most common one. People are having rashes, fatigue, nausea, and stuff. And this varies from one vaccine to the next. The Pfizer vaccine, which has been used the most in Canada, has the most. And when they say serious side effects, they mean anaphylaxis. People get allergic to it. And you can see that from Moderna and then for the Covishield, which you've given less of. To date, over 806 million people worldwide have been vaccinated with one of these vaccines or another. And essentially what you're trying to reach is what we call herd immunity. I want you to watch this animation as I wrap up. This is what happens when so here you have people who are mostly unvaccinated. So you don't have any vaccinated people here. You have 20%, 40%, 60%, 70, 80, 90% vaccinated. And the idea is that we need close to 90% of people vaccinated so that it can be a breach for the virus so that it can't infect others. So you can see when nobody's vaccinated, everybody gets infected with a highly contagious virus. 20% of people vaccinated, negligible amount are spared from being infected. 40%, 60%, and until you reach about 80 to 90%, you're not getting that level of protection. A vaccine like measles virus vaccine is about 99% efficacious, right? And so you need 95% of people in the population to be infected to protect you from measles, which is a way more contagious virus, by the way, than your coronavirus. So what we've seen so far is that we've seen that the vaccine has worked. Here, they did a study in Texas of hospital workers who were vaccinated and compared those with the rate of infection versus the unvaccinated group. And here you can see for people who are not vaccinated, 200 and something of the 8,000 something got infected. Here are people who are partially vaccinated, one dose, and you can see that that number already is reduced. But look at people who are fully vaccinated. Only 4 out of the 8,000, so 4 versus 230 something over the same period of time. So in other words, the vaccines actually work and work pretty well in protecting people from SARS-CoV-2, despite of the fact that they have these side effects and even some of the current issues that we have. It was a convergence of luck and preparedness. As I showed you, they were just going into phase 1 and 2 clinical trials with these. Moderna was already working with the NIH, National Institutes of Health, where a young African-American woman, Cosmika Corbett, has been working on coronavirus vaccines for over 6 years. And essentially, at the time when the pandemic hit, having tried this platform, having sequenced the spike protein and knew all the structure of it from previous coronaviruses, they decided that, hey, there is no time like now. And several things came together. Number one, Moderna and Pfizer and BioNTech, so Pfizer actually partnered with BioNTech, are small companies. They literally were started by professors who were discovered this mRNA technology. They did not have the money or the investors to push to make a vaccine at this rate, but federal money became available. So they were able to try the platform. Number two, because we're in the middle of a raging pandemic, they had all the tools and all the assistance from all over the world that they needed in order to try this technology. They had already established safety. They went back and they did the animal trials again. They were able to go into a phase 1 clinical trial almost immediately, within 2 months of getting the sequence of this virus. They were able to go right into phase 1 clinical trial to see if the vaccine was safe in humans. Again, remember, they had already established safety for other phase 1 trials that they were doing. We didn't pay attention to it because Zika and chikungunya and some of these viruses don't concern us. They affect the Caribbean and parts of South America, and we didn't have to worry about it. So we didn't notice that they were testing it. And so this technology was ripe at the time. You could call it luck that it was there, but it all converged around the same time, and they had the resources that they needed in order to go forward and test it. Remember, they didn't know that it would work either. There was a strong possibility that it might not have worked. But as they continued through the trials, they got better and better results. It's simply not the case. And here's why. Again, we've seen, when we talk about the sequence of the virus, in other words, the entire genetic alphabet that makes up this virus is publicly available. Because it's publicly available, as a scientist, I can go and download it and look at the sequence. There's software out there that you can use to run and look at the code. Now, human beings, as a microbiologist, I have the tools, the equipment, and the expertise to manipulate an infectious microbe. I can make it worse than it is. I can make it less infectious. But here's the thing. The tools that are available to do that are widely known. We have to use what are called plasmids. We have to use certain enzymes that cut out specific sites. In other words, if somebody had modified this virus, the signatures would be there in the sequence. Those can't be hidden, right? And we would be able to readily, all the scientists over the world who are looking at this, we would be easily able to say, hey, this thing was manipulated. Now, is it a possibility that somebody had this working in a lab and somehow somebody got infected and that came out? It's always a possibility when you're working with an infectious microbe for somebody in the lab to get infected and then take it out. However, we have seen no evidence that that has happened either. What we know from these viruses is that they're constant in jumping from bats to humans. We've seen it happen repeatedly. So what we have the opportunity to do now is that we can believe that something nefarious happened, or we can believe what we've seen repeatedly over the past 50 years happen with this family of viruses, that they jump from the animals that they typically infect and don't cause death and disease to humans because we continue to encroach on these animal habitats. The sequences that we've seen shows that this is coming directly from a bat. So that's the closest sequence we have. And then there are sequences that suggest that it might have gone into an intermediate animal called a pangolin. And they continue to search for other sequences to see if it matches closely to anything else. It could have jumped from one animal to the next, and then from that animal into human as it acquired the ability to infect humans. But there is absolutely zero sign right now that this virus was manipulated. In other words, weaponized in a lab and then sent out. That absolutely did not happen. Initially, we didn't know how effective these vaccines were against different variants, but we do now because the research has been done. And what the research has shown is that both Moderna, the Pfizer, the Johnson & Johnson, and the AstraZeneca vaccine shows very good efficacy against all of the variants. And we have to be happy for that. How do they do that? They were able to figure this out by taking the sera from people who got the vaccine and mixing it with the viruses and see if the virus is able to escape. And it has been able to effectively escape. Now, is it as effective against the variants as it is against the initial strain? No, it's not. Against the initial strains, especially these mRNA vaccines are 90-something% effective. Against the variants, that number drops to 80-something to 70%. Now, here's the thing. If your vaccine is 50% efficacious, it's going to get approved by the FDA. So it is still way beyond the threshold for approval overall. I'm not concerned about the current variants that we have, that they will escape the vaccine. What I'm concerned about is the next variant that is probably brewing somewhere in the world right now that we haven't yet seen, that could completely escape these vaccines or be so to make these vaccines less effective. But for the variants that we have now, the major variants that we have now, the vaccines are pretty decent against them. All of these vaccines, they prevent hospitalization and death, which is really what you're looking for. We're not going to need to vaccinate annually like the flu vaccine because this virus is different. You know, for the flu viruses, they change their outer surface so much that the neutralizing antibodies made by the system doesn't recognize them. So they completely escape or almost completely escape. This virus doesn't make half the mistakes that the flu virus make because it turns out that it actually has a proofreading mechanism. It's like when you're typing a paper and you put a spell check on, this virus actually has a spell check. However, as you know, even when you have a spell check, you still make a few errors here and there. And if you have millions of people being infected, those errors add up to a lot of errors. And that's why we're seeing variants. So if we cut the number of people who are being infected today, the number of variants will go down, or the probability of getting variants will go down significantly. So if people continue to wear their masks, social distance, wash their hands until they get vaccinated, vaccination is not what's going to take us out of this problem because we can't vaccinate everybody at the same time. you will actually be able to reduce the amount of variants. But no, we won't need a booster every year. The only reason why we would need a booster is if the amount of antibodies wane, or if we have a variant that this vaccine doesn't cover, and we haven't seen that yet. No. So, it would never be equally effective because the vaccines, especially the mRNA vaccines, were made against the original strain. In fact, when the mRNA vaccines were being made, nobody knew that there were other strains available. But since then, what they have done, both Moderna and Pfizer have gone back ahead, and they are now in the process of creating a vaccine against all of the variants that they see, right? So that's the beauty of the mRNA platform. It's like a plug and play. So it's very easy to do that. And so they're in the process of actually doing that right now. We won't need it yet because, again, we're not being overrun by strains that are evading the vaccines. People who are vaccinated, we see them resisting the B117 strain. We see them resisting the 315 strain. We see them resisting the P1 strains of the virus. However, if there turns out to be people who have not yet been vaccinated in areas where those strains are the major ones, maybe when this vaccine, the new strains of the vaccine, the new variants made in the vaccine are available, those people would be vaccinated initially against those. And we would only get like a third shot if one of those variants was evading and was overrunning a certain locale. I've seen these videos go around, and to be honest, Adventists have become some of the worst perpetrators of what the anti-vax movement have been trying to do for a long time. What seems on the surface as this innocuous little thing that's being done by a few people here and there, it turns out that it's a bigger machinery. The anti-vax movement is such a well-coordinated one that most people don't even realize when they're being pulled into an anti-vax argument and situation. Anti-vaxxers don't make up a large portion of the population, but they're extremely effective in playing on your fears, on your religion, on specific cultural beliefs. They find these loopholes and they tie in. I just showed you in this presentation what is actually in these vaccines. In fact, for these mRNA vaccines and these viral vector vaccines, they're probably the safest vaccines and the cleanest vaccines we've ever had in our human history. I mean, when you think about it, in 1796, when the first vaccine was given, it was given from a pus, from a cow, to a boy to be able to prevent smallpox. You can imagine that in that pus, you had immune cells, you had bacteria, you had, yes, the virus, the cowpox virus, that was an attenuated strain of the smallpox, but you had so many things. That vaccine would never have even made it into clinical trials today, right? Today, we have the technology to test everything that's in a vaccine. As I showed you, what the vaccine has in it, it has mRNA, it has lipids, and it has sugar and salt, right? Tiny amounts. There's no formaldehyde in it. But these people don't care that there's none of that in there. They don't really care that what they're saying is not true. And you might think, well, but these are Adventist church members. What has happened is that they've been put on a path where they have to purposely now turn around and ask themselves, if I can believe all of this stuff that is coming out, why am I not believing the people who actually work with this stuff? Why am I not believing the people who are experts in this and who have looked very closely at it? You have to ask yourself that. If you want a balanced conversation about it, look at everything. If you look at everything, you're going to come out with what I just, and that is why when somebody invites me to speak on this, I don't come and just sit in front of my computer and talk to you. I put these slides together for two reasons: that I love illustrations, but if you notice on every slide, I show you the source where it's coming from. And I show you the scientific evidence that backs up what I am saying. So now it's not my opinion. It's just me interpreting the science for you and trying to break it down so you can understand it. And that is why I give these presentations in this way. I don't just go on a platform and talk about these things. I use science-based evidence because I feel that if I can teach you the science, I don't make any assumptions that you can't learn the science. If you can learn the 2,300-day prophecy, you can learn the science of this. And if you understand how basic cellular responses work, if you understand the basis for vaccine, then a lot of these things that people take to you, you'll immediately know that they're false. And I hope that to some extent, I have prompted you this morning for you to be able to do that. And so what they've said is completely false. These vaccines have none of those things in them. Ellen White herself took the smallpox vaccine because it was the smart and right thing to do at the time. She probably would have died from that disease if she hadn't taken it. Yesterday morning, I was reading in the book of 2 Timothy, and it's like this text just jumped out at me, 2 Timothy chapter 4, verses 3 to 5. If you can go back and read that. And it says, "For a time is coming when people will no longer listen to sound and wholesome teaching." And we often think of that of just being the gospel. They will follow their own desires and will look for teachers who will tell them whatever their itching ears want to hear. They will reject the truth and chase after myths. But you should keep a clear mind in every situation. Don't be afraid of suffering for the Lord. Work at telling others the good news and fully carry out the ministry of God that God has given to you. That's the New Living Translation of 2 Timothy 4, 3 to 5. It's very easy to get caught up if we're not really analyzing and effectively parsing what is passed along to us. The variants that we have now, the P1, the B117, all of these are variants that developed in the human population. The virus itself is itself a variant, because remember that it's a zoonotic virus, right? In other words, what it typically infects are animals, and it lives okay with them. They have an agreement, they have a lease agreement where it lives in these bats. If it doesn't get out of control, then the bat doesn't bother with it. It can get its food, it can get to replicate itself, because viruses don't want to kill us, you know. It might feel like that's not the case now. All microbes, their only goal, they're programmed genetically to make more of themselves. They're progeny oriented, right? But they will make progeny at the expense of your life if that's what it takes. That's how we end up dying from it. So all of these variants that we're saying, if you notice, I'm not even saying strains. They're variants because they're so close to the original thing that you probably would not be able to distinguish them unless you completely sequence the entire code. And so it is not the case that if it's coming from animals, then you don't have to worry about these variants. But if it's in humans, then we have to. The fact is that if we're building vaccines like the ones that we have today that are able to neutralize those. So when you make a vaccine against a spike protein, remember I showed you in that presentation that there were those 3 black dots on the binding region of the spike protein for this virus, and that's what made the B117 variant. Now, when your immune system sees the spike protein, it doesn't just see that binding area. It sees the entire spike protein, and for the immune system, that spike protein looks like a 10-story building, where the immune system is making a response, making antibodies against every window, every door, every ledge, every part, right? And so the virus might change the shape of a window ledge here and a door there and something else there, which makes the building look a little bit different, but not enough that what is built against all the other things won't respond to it. I hope that makes sense to you. It's what we call a polyclonal response. It's not just responding against one tiny part of that protein. It's responding against every stretch of 8 to 10 amino acids. Is another epitope that the immune system sees as discrete and makes a response against. So while if you make a response against the entire 10-story building, it's better. Making a response against 75% of that 10-story building is also effective and can also prevent you from hospitalization. That's very possible, very possible. Hopefully not, but very possible. And the only thing that can stop it is if people stop getting infected. And we have a lot of control over that right now. If we social distance, if we do the things we're supposed to do, and if when people are eligible for the vaccine, they take the vaccine, then that helps to slow the spread. And if you're not having the spread, you're not having the mutants developing. Oh yeah. These vaccines for the coronavirus only protect you against coronavirus. It does nothing for all of the rest of viruses and bacteria and other stuff that we have in the population. So people still need to get their flu shot. However, if you notice what has happened in North America and in other parts of the world over the course of this pandemic, that the social distancing, the wearing of the mask has significantly reduced the influenza. In fact, we really didn't have an influenza season. I think we had a single pediatric death from influenza in the United States this year. Typically you have tens of thousands to hundreds of thousands of people dying from the flu at this stage in the season. We just haven't had a flu season. So I don't know what that's going to say about us moving forward, Director. If people are going to say, in flu season, you probably should wear a mask, but for sure, you're going to still need the flu shot. And for people who work in healthcare, for example, the hospitals don't allow you to work there unless you've gotten the flu shot, because of course, it's also one of those very contagious viruses that you can spread very rapidly. So the current virus, SARS-CoV-2, is extremely similar to SARS-CoV-1, which is the 2003 variant of the virus. They're similar viruses. This one, however, binds much more tightly to the ACE2 receptor, is way more transmissible, has some variations there, especially at the binding domain in the sequence. And it binds to the ACE2, but this current one is also able to bind to other receptors to be able to get into a cell. We haven't talked much about that. The immunobiology of it is really interesting. It's developed this enzyme using the cellular enzymes to cut and to help the virus to fuse and get in. And that's way better than what the 2003 virus actually has overall. Right, we've never had an approved mRNA vaccine. In other words, nothing has gone up to the FDA or to the MHRA in the EU to be used as a vaccine before, because it was in trial. But yes, it has been used in humans before in terms of trials, but not as a licensed vaccine. Where long-term risks are concerned, you might not immediately realize this, but vaccines typically don't have long-term consequences. For the very reason that I've told you. In fact, what the FDA did when people started bringing this up is that they went back and checked for the last 50 years of vaccines that have been approved. And what they saw is that they had no case of an adverse event from a vaccine after 2 months of that vaccine being given. Why? Because these vaccines are typically proteins. As I told you, once the mRNA is read, it's degraded, right? That happens with our own system. mRNA is created, that copy, to make the protein. Once it's read and that protein is made, that mRNA is gone. So the mRNA is not sitting around in your body for days, weeks, months. No. It's gone within hours of it being read and it's done with. The protein that is made from it is also gone because the protein has to be degraded. It has to be broken down into its amino acid component for the immune system to effectively read it, especially the adaptive immune response. The adaptive immune response is highly sophisticated and it reads amino acid sequences, right? It doesn't read the whole structure of the protein. So that's also degraded and broken down. So in my estimation, and I haven't seen the research for this yet, within 2 to 3 weeks of you getting that vaccine, if I should go and test your body for the presence of the messenger RNA or for the spike protein, I wouldn't find it. It'll be gone. So in terms of long-term consequences, any long-term consequence you're gonna have would actually be related to your immune system's response to the entity and not the entity itself, the vaccine entity itself sitting around in your body, because vaccines just don't do that unless, of course, these vaccines are attenuated vaccines. Like the MMR, the mumps, measles, and rubella vaccines are all attenuated strains of those viruses. In other words, they're giving you a live virus that is able to stay in the body for some time. And sometimes those viruses actually get in the intestines. Polio as well does that. The Sabin polio vaccine gets into the intestines, replicate, and that's how your immune system ramps up against it. So sometimes if your immune system gets damaged, those attenuated viruses or weaker viruses could actually gain strength and cause problems. However, none of these, what we call subunit vaccines, pieces of protein being given, pieces of message to make a protein being given, none of them actually stick around. So scientifically, from a scientific perspective, I wouldn't expect to have any long-term effects of a vaccine like this because there's just nothing there. Very good question. At this stage, both not just the AstraZeneca, but also the Johnson & Johnson, they're built on the same type of platform, different viruses, but same platform. And essentially what is happening here, here's the issue. When you do a clinical trial, you do it on tens of thousands of people. So the AstraZeneca and the Johnson & Johnson vaccines were made, and the phase 3 clinical trial involved 40 to 44,000 people. These clotting events that you're seeing are events that they see maybe 1 in every million person. So you wouldn't have picked that up in a clinical trial because you just didn't have enough people. Right now that they're giving it more broadly, as you saw 5-6 million people in Europe, and they had 18 or so cases or 22 cases of people with these rare blood clots. What they need to do now is that they need to go and figure out, is there a certain underlying condition that people have that predispose them to having clots when they get this vaccine? And then those individuals need to be excluded from getting the vaccine. In any vaccine, over time, you have groups that are excluded from getting vaccines. You have people who are excluded from getting certain drugs. That part of the advertisement that they do for drugs that they go through really fast, those are people who are not supposed to get it, and adverse events that it causes. So them pausing for a little while and also not giving it to people who are younger, because the younger you are, is the less likely it is that you'll be hospitalized or die from COVID. So the risk, vaccines are a risk-reward ratio. The risk of you developing this thing, this rare clot, is too high compared to the reward of you not being hospitalized. And so I agree with the places that have pulled back on those vaccines at this time while they investigate, and lots of people are on it right now. My hope is that they'll find either a genetic underlying or some other condition that is leading to this, and they'll figure out. But what that says to me, it says that the process is working. You're still doing what we call in vaccine development a fourth phase of clinical trial. In other words, after you've given it to the general public, you continue. In fact, you're mandated by the federal government to continue to examine if there are adverse events that are unwanted or were never planned for. And certainly these rare blood clots, are unwanted and unplanned for, and it needs to be figured out. So I'm all for that. It all depends on where you are and then the availability. So for example, when I was getting vaccinated, I could choose which vaccine I got. So for example, as you know, for example, in Jamaica, they only have the Pfizer. They have a single vaccine that they're getting. In the UK, in the US, in Canada, for example, in Canada, you have 3. And you would be more than likely to get the Pfizer because Canada has decided that it's restricting the people who are getting. You're more than likely to get the Moderna or Pfizer vaccine at this stage in Canada. What I would say to that person, I would say, how could you be so cold as not to notice that almost 3 million people have died? You see, it's all about numbers. You see, people say this as if it's really a thing, that, only 2% of people who get infected with this die. Are you okay with 2% of your congregation dying? Are you okay with 2% of possible people who need to be evangelized dying? What is an acceptable number of deaths from something that's preventable? So that's number one. It's all about numbers. When you have over 136 million people infected, 2% of that is a lot of people. If you're talking about just 100 people, that's only 2 people. But once you go up in numbers, it gets exponentially higher. So that's number one. Number two, these variants that are going around, what we're seeing now, initially, only older people were ending up in the hospital. Now the older folks are getting vaccinated. What are we seeing now? The vast majority of hospitalizations that we're having here and that you're having there, I looked, are younger people, people under the age of 50. because the B11 strain is that much more contagious and cause severe illnesses in younger people. Here's the thing, none of us know what is it that we have that will cause this virus to infect us and make us end up in the hospital. And the third thing is what I showed you at the very top, that even people who survive have what is called long COVID. So the morbidity, we talk about mortality a lot, but morbidity for people in epidemiology is a really important thing. This thing might not kill you, but it might debilitate you so much that you're not able to work. You're not able to carry on with your normal life. And as I told you, there are people who have committed suicide after having recovered from COVID because the symptoms, the long COVID symptoms, are so terrible that they can't overcome them. So the question that somebody now has to weigh is, what do I do? Do I roll the dice with something that has the potential to cause such great mortality and morbidity? Or do I simply do something that we've been doing for over 200 years, take a vaccine? That's really the choice. It's a loaded question because it depends on so many variables, right? It depends right now what the rate at which we're vaccinating here in the US is going to drop significantly because the pace was dependent on the Johnson & Johnson vaccine. Same thing for you in Canada. Your pace, the rate of vaccination has slowed down because you were depending on the AstraZeneca vaccine as the third vaccine to build that stock, and you're not vaccinating a whole swath of the population with that. So at this stage, unless the vaccination rate is ramped up significantly, at this stage, demand is outpacing the production of these vaccines. So it's not clear to me how long this is going to take. It's definitely going to take beyond the summer, though, unless something new gets approved and starts being used in high amounts. In terms of lockdowns, I think once you reach a lower rate, right now you're surging. In other words, you haven't even plateaued there in Toronto, you're still surging. And so the government is not going to move those restrictions until there's some leveling off and falling off of those numbers. But I think once those numbers have fallen back to a reasonable amount, even if people are not vaccinated, you will be able to go back to worship in some way. And in fact, last week, my congregation here in Springfield, Massachusetts, was able to go back to church for the first time since last March. We didn't fill the sanctuary, but we had people in there. We were still doing a live broadcast part of it so that people at home could get it. We had a sign-up sheet, did a Google sheet where people signed up. They told us whether or not they were vaccinated. The vast majority of people who came, some of the older people who were most affected, being home alone, those people are all vaccinated. They have their 2 doses of vaccines. And so they felt more comfortable coming to church. They were very happy to be in the sanctuary. But in our community, our rate is less than 2%, right? And so our mayor has said, and our governor of our state has said, if the rate is above 2%, then you have to follow these restrictions. So sooner or later, some of those restrictions will leave. However, where the mask is concerned, we're still going to be wearing masks because everybody at church wore masks. Everybody around, even those of us who are vaccinated, continue to wear masks. Why? Because of these variants that are known and that unknown, that variant X, that we haven't yet known about. You have to be concerned about that. Remember, vaccines are not 100% protective, right? So while they're really good, even the ones that are really, really good, there's still that 5% chance that you'll have what is called breakthrough, where you could get infected with enough dose of a variant that break through the vaccine and you end up in the hospital and could potentially die. So until the numbers in the community go down significantly, the numbers of infections and the rate of vaccines go up, then of course, we should continue to wear those masks and social distance and be safe. I'm not a pharmaceutical company, so I wouldn't try to rebuff it. But when you think about R&D for a vaccine, you're talking about billions of dollars, not millions of dollars. You're talking about billions of dollars. Who do you know out there? When was the last time somebody in our church published a book and could get it for free? And you have to buy it. So I think we would be naive to think that a company would license the technology to produce a vaccine, pay millions and millions of dollars for that licensure, have a production system in place with tons of factories worldwide producing this thing, having employees that have to be paid, working around the clock to produce a vaccine, and one wouldn't have to pay for it and that they would make a profit. It is the profit that they make that actually allow them to do what we call R&D, research and development, that is in place so that the next thing that comes, they might be on the cutting edge of trying to figure out. Remember, the technology wasn't developed when the pandemic came. The technology was already in place. Who developed the technology? It takes money to do that. So I'm not cynical enough to think that vaccine companies shouldn't make money. Neither am I naive enough to think that they're not making money. You know, it all depends on what they decide to charge for a single vaccine. And that's why it's so sad that the AstraZeneca vaccine and the Johnson & Johnson vaccines are having issues, because it turns out that they're the cheaper ones on the market. Not necessarily because they're the cheaper to make, but the manufacturers have made it so that they're cheap. So, you know, we have a lot of drugs out there, a lot of stuff out there that has to be bought. And we should expect that, you know, we live in a consumer-driven society and companies are going to make money off their products. If we came up with something that was effective in whatever way, my point is that, yes, they can make money from it. Yes, there is some person with stocks in these companies who just became a millionaire or millionaire multiple times over, but that shouldn't prevent us from getting the vaccine. And it is not the case that all that is happening is that these companies are getting rich off a vaccine. There's a market for the vaccine because we have a global pandemic. I hope that we don't have nationwide or global vaccine passports, because what that would mean is that it would force people to get vaccinated. I'm a proponent of safe vaccines. I'm not a proponent of bending people's will so that they will take a vaccine that their conscience does not want them to take. I mean, I think that's violating a lot of people's rights. However, to a great extent, vaccine passports already exist for several things. In many school systems, if your kid is not vaccinated, they're not going to school. As I told you before, if you don't get vaccinated against the flu, you're not working in several healthcare settings. My wife is a healthcare provider. She has to get her flu shot every year. Otherwise, she can't work at the hospitals and the nursing homes that she works at. And so to a great extent, that is already a vaccine passport. We now have a COVID test passport for most countries. If you don't test negative, you can't board a plane to go to certain places. So I think I would be naive into thinking that we won't have companies, countries, regions saying, hey, unless you can show me that you've been vaccinated, and there are a number of companies that have already come out and said that they're going to ask people to vaccinate and they're giving you a certain amount of time to do it. And if you're not vaccinated, you'll lose your job. That's going to happen. And for private entities, that non-government entities, people are going to do that. I still believe that a lot of the people who don't want to get vaccinated is not because they're anti-vaxxers, it's because they're vaccine hesitant, and that our government needs to do more in educating people about the safety of these vaccines, the science behind these vaccines, why you need to take it, rather than laying down a hammer and saying, if you don't take it, then you're going to lose these individual rights. And I'm hoping that it will come out on the latter rather than the former. I laugh when I see that. And the reason I laugh when I see that is that the very people who are saying don't get the vaccine are the same people who are saying, hey, why aren't they giving us ivermectin? Ivermectin is a good drug, but it's not an antiviral drug. Ivermectin is an anti-parasitic drug, right? It was designed to treat parasites, and it's really good at it. It wasn't created to be an antiviral drug. Now, there could be off-label uses of drugs, but all of the tests to date have shown that ivermectin doesn't have any efficacy against COVID-19. Where did people get this idea that ivermectin could work? A single publication by a group that they did in cells and tissue culture, not in human testing, in tissue culture, where they used somewhere in the order of 100 to 160 times the safe dose of the drug on cells. and saw that there was an effect on the virus in tissue culture. Now, as somebody who grows cells and infect them and treat them with drugs, I can tell you that sometimes what happens is that the drug itself make the cells so sick that they don't get infected with the virus. And you could take that to mean that the drug is killing the virus. You see that the virus are not going in the cells because the cells are too emaciated for that to happen. And so people need to be careful and cautious. You can't take a drug because you say it is safe for this type of disease and just throw it on and say, hey, since there's a safe drug. They did the same thing for a hydrochloroquine, which we know now has no efficacy against this virus. So people should really stick with what is known. And that, however, is an area that is missing in COVID-19. You have a vaccine. What we don't have are drugs that can treat people pre-hospitalization so that you don't end up in the hospital. And I know that there are a lot of efforts right now looking at that to find drugs that are reasonably good to treat the virus. However, this virus has a proofreading system, and a lot of the drugs that you would typically use to treat something like an HIV or even Ebola won't treat this virus, because since it has a proofreading system, what we typically do is that instead of it picking up one of the DNA bases, the ATGC, we give it a poisonous base, call them nucleoside inhibitors. And that's how you typically kill viruses. You give them a poisonous base. They don't know that it's poisonous, so they take it up and add it to their genetic code. And when they do that, it terminates the viral nucleic acid from happening, so the virus dies. This virus has a proofreading system. So when you give it a base that is a poisonous base, it doesn't pick it up. Or if it picks it up, the proofreading protein cuts it out. And so a lot of the drugs that would have worked against it are not going to work because it has this extra thing in there. And the reason for that is that it's a large virus. In terms of viruses, this is a pretty large virus. That's 30 proteins. Most viruses, like even the flu virus, has like 8 proteins. HIV has a handful of proteins. This virus is a large virus. It's acutely aware of its environment and is able to evade in that way. The AstraZeneca vaccine is just manufactured in different places, but it's the same template, right? It's the same plasmid that they just ship to India or ship to other places that are able to manufacture it overall. So it's pretty much quality controlled from the development stage. Yes, I would recommend it because yes, there are rare events, but those events are so rare. And especially if you're in an age group above age 50, I definitely recommend this over you not getting a vaccine at all. It's an efficacious vaccine. It has been proven repeatedly to prevent you from getting hospitalized and dying. In fact, it was tested against many of these variants. The Moderna and Pfizer weren't because they were produced earlier on. So this is still a good vaccine. There are lots of people who have gotten this vaccine and have been protected. The data has shown that people have been challenged with the virus in the environment, didn't get sick, didn't even realize that they were infected after having gotten this vaccine. However, I'm still concerned about these rare blood clots. And I'm really looking forward to them finding the basis for these. And until that time, I think people in the younger age group probably should avoid getting, since that is the age group, especially women, that we're seeing the vast majority of those clotting episodes in. Again, you're talking about a handful in millions of vaccines, but it's enough to have some caution there. COVID is not diagnosed by a symptom. You don't get a COVID diagnosis until you've tested PCR positive. So you can't mix up COVID and the flu where that is concerned. The reduction in the flu, as I indicated, is because we're social distancing and wearing masks. So I don't know of anybody who's being misdiagnosed with COVID having had the flu. Number one, we haven't had much flu. But number two, the basis for COVID testing, 99.99% of all COVID testing is nucleic acid based. In other words, we're not just seeing the body's response to it, we're looking for the presence of the virus. I'm looking at 2 different genetic codes in it. So very little is left up for chance there overall. So that's the way I would answer that question. Yeah, the vaccine has not been fully tested in children. We know that the initial data looks good. For Pfizer, Moderna is doing the same thing. Pfizer is applying for emergency use authorization to the FDA right now. They're getting ready to do that. So 12 to 15-year-olds will be vaccinated. One of the things I didn't mention is that the ACE2 receptor, that protein that the virus binds to enter our cells, it turns out that young children don't have a lot of it on their cells. It's the hormones at puberty that drive up the level, both progesterone and estrogen. So until a person reaches puberty, the numbers of those receptors are low. It's not that you can't get infected, it's that the probability of getting infected is so very low. So for children, as is the recommendation now, they should still wear masks. They should still social distance as much as possible so that they don't get infected. You know, school every day. He's in school right now. He wears a mask. Social distance, has been doing it since last fall. Hasn't become infected. So it works, the social distancing issue there. So I would say that's what we need to do right now. Even if a vaccine becomes available, it take a long time before all kids would actually become vaccinated. They still have to test it. They're right now testing it in kids 6 months old and up as well to see. So eventually, I expect that this vaccine is going to be tested on all age groups so that even young kids will be able to get it. But don't expect that COVID-19 is going to go away. Even after we're all vaccinated, this virus is still going to be around. You're going to look at the news one evening and you're going to see somebody else who died from COVID-19. It'll be rare, but this virus loves us too much to let us go. It binds so tightly to our receptors. It's so easily get transmitted that it's not going to let us go. In 1 Corinthians 12 and verse 7, I could even go from verse 1. It says, now about the gifts of the Spirit, brothers and sisters, I do not want you to be uninformed. I'm going to jump down to verse 4. There are different gifts, kinds of gifts, but the same Spirit distributes them. There are different kinds of service, but the same Lord. Verse 6, there are different kinds of workings, but in all of them, in every one, is the same God at work. So verse 7 now. Now to each one, the manifestation of the Spirit is given for the common good. To one, there is given the Spirit of a message of wisdom. To another, message of knowledge by means of the same Spirit. Verse 9, to another, faith by the same Spirit. To another, gifts of healing by that one Spirit. I want to pause there on that gifts of healing. If you notice, all of the others is like gift, but there are very few in this list that are gifts of healing. It's unfortunate that in a church that has the health message, a lot of our members haven't realized that we have the gift of healing. And it's not some miraculous Wednesday night prayer meeting. It is the professionals that God has given wisdom to, the doctors, the nurses, the scientists that create the tools and the means by which these physicians and patient-facing individuals can actually work. That's why it's the gifts of healing. It's not a single gift of healing. It's not a single type of healing. It's the gifts of healing. And I would like to think that as a scientist, as a Christian scientist who devote my life to finding treatments for chronic and acute diseases, I believe with all my heart that I'm doing the work of God. And when you tell me that I'm not, that I'm doing the work of the devil, that every vaccine and every medicine is evil, I say to you, didn't the God of wisdom give you that gift for you to decipher what is good and what is not? And haven't you read that not everybody is going to be a minister? Not everybody is going to prophesy. Not everybody is going to teach. Some people are going to heal. And I hope that you see the development of these vaccines. No human being can come up with this stuff out of the blue. I tell people the more I study the immune system, is the more I agree with David that we're fearfully and wonderfully made. What I just gave you is a synopsis, but when I talk to you about a vaccine and about a microbe coming into the body, I'm picturing every cell that is responding. I'm picturing every communication of these cells, every protein that is produced to tell that cell that it's time to come in. This gene is upregulated, and then that system releases itself. This cell over here is maturing. This is being acutely upregulated and disseminating its products in an effective way so that you can be protected, because that is the gift that God has given me, for me to understand at the molecular level how this body that he's created works. So for you, based on your knowledge, when you think about God getting down in the garden and creating man, you see God just taking up a bit of soil. For me as a scientist, I see him looking for a specific patch where the right mineral content is, and using that to create specific organs, and using that some parts to create the skin, another part to create this. So it's very specific based on the knowledge that I have. And I hope that from time to time, we will expand our knowledge base to include just this underlying wisdom of God to create this body in such a way that even after abuse upon abuse, it still functions in such a way that I marvel every time I discover something new as to how this system is designed, layer after layer after layer, to protect us, that even after we've stripped away those layers based on our lifestyle and our wrong attitudes, we're still protected. Vaccines don't work on their own. Vaccine is just an entity that you put in the body. It is the immune system that God has created that does all of the rest. Antibiotics don't cure your infectious disease. We only get disease because the immune system is overwhelmed. What antibiotics do is that they take the number of microbes down to a manageable level, and it is the immune system that clears the infection. Antibiotics cannot clear an infection. The immune system does. If you understand it from that perspective, then you have no choice but to say, thank you, God, for creating me with such intricacies. And thank you, God, for giving the gift to people who can understand this intricacy. Because if you don't understand how a car works, you can't fix the problem. The people who are creating these fixes understand fundamentally how the body works. And that's the way we think. So when you come and you say, hey, the vaccine is doing this and it's doing that, we don't know what you're talking about. We can't understand how you could stick with that point when all clear evidence shows that is to the contrary. I pray that God will continue to bless you and your ministry. You are so important in taking us out of this COVID. Lots of us are going to need a lot of counsel. We're going to need mental health help. And it is from the pulpit and from your lips. A lot of us are going to get this therapy because we're either too scared or don't have the resources to go for it other places. It is therefore important that you understand what you're saying, that you understand the fundamentals of what is going on. And I'm here, if I can ever help, I am on hand to help you to understand this stuff. And you can go to other professionals who have other expertise in other areas, but this is mine. God bless you.